Cheyenne and/or Miles, can you give me a thumbs up if it looks good, please?
Yeah, I see it on my end.
Okay. Okay. Thank you. Thanks, Miles. All right, everyone. Good morning. Welcome to another fine seminar. My name is Lauren Hayes, and today I have the pleasure of hosting today's seminar. Just a couple of announcements before we get started. First off, I want to just highlight last week's speaker, thank Doug Emlin for a wonderful talk, and I'm going to share my screen to show you the link to the website. So the last week was Doug Emlin, and let me see if I can find this. And gave a great talk on sexual selection and duels. Fascinating talk. Next week we have Christian Perk, who will be from the University of Pretoria, talking about pheromones, diet, and reproduction in the honeybee. And I wanted to just quickly announce that there are, this website that you see here, we've developed, the co-coordinators developed in collaboration with Facundo Fernandez Duque, where we are now starting to build resources, particularly related to teaching. And so I want to click on this really quick. These are the links to different resources that we're preparing, including, for example, if you click on teaching slides, you can see that we have uploaded various teaching slides that you can download for your classes if you're interested. And we're soliciting more from our fine speakers. We also have the certificate of participation if students need this to get credit for attending a fine. All the reading lists are here. And then we give some recommendations for how to use fine in a classroom. I basically, for this part of the site, I'm showing you what I do with my behavioral ecology students. So hopefully we're going to be building this up over the coming months, and hopefully we can have a nice resource for people to use, for their teaching. Let's see. So just for the new folks here, just a reminder about the structure of fine. We have about a 45-minute presentation, after which we have upwards of an hour of discussion, question and answer discussion. We ask that during that question and answer, one of us will be moderating that. We ask that you put a question mark into the chat, and we'll call on you as in the order they come in. We are trying to encourage some of the juniors, people here, especially students and postdocs, to really get involved. So please ask questions. We want this to be an opportunity for you to get to interact with the speaker. And it's regarding the YouTube page. I've got people monitoring that so that if you have questions there, they'll be sent in to the moderator so that those of you watching on YouTube live, you can get your questions sent to us. And so with that, really, really happy to introduce our speaker for today. It's my pleasure to introduce Joan Strassmann. I just had a nice chat with Joan a little bit before the seminar today, and she basically reinvigorated me to want to participate today. She is a Charles Rebstock Professor of Biology in the Department of Biology at Washington University in St. Louis in the United States. And I'm really excited to have Joan give the seminar today because she's a leader in animal behavior. And from my readings, I realized that she really sees the big picture. She sees the big questions, and that's really exciting. She's received obviously a number of awards. She's a National Academy of Science member, Guggenheim Fellowship, Fellow of the Animal Behavior Society, and she was also past president of the Animal Behavior Society, which says a lot about her service to the field. From her website, which I encourage everyone to go take a look at, she describes her research program as investigating cooperative alliances that have occurred at several important steps in the evolution of life and have proven evolutionary and ecologically very successful. Her interests are largely in how, and I found this really, really fascinating, how conflicts are subsumed into cooperation, an idea that I really, really have enjoyed learning more about. And this has been sort of the main focus of her work in the past few years. She's been largely focusing lately on the evolution of cooperation and conflict. And I apologize for the cardinal that's calling right next to me. It's quite loud this morning. The evolution of cooperation, conflict, and social amoeba in Dictyostelium discoideum. I'm sure I butchered that. Social amoeba. But in addition to this really fascinating work, she's done a lot of work previous to that in social hymenoptera. She's also done some more technical work like developing microsatellites. And I guess I came to the opinion as I was learning more about her that her work is really impressive because, as I said earlier, it reveals the big picture issues. And her thinking spans from levels of complexity from genes all the way to whole organisms. And I think that's really something that is exceptional. Not surprising, she's been very productive, well over 200 papers in journals like Nature, Science, PNAS, Biology Letters, and all the behavior journals. And her work is well regarded. She's been cited over 14,000 times. But one of the things I learned from her today, and having read her website, is that she's also heavily interested in education, teaching. She's supported, from what I can tell, well over 25 PhD students, over 20 postdocs. And really, she maintains a sociobiology blog that I think is a good read for everyone. Early career, mid-career, late career, wherever you are, it has some really sound advice on things ranging from getting students to write, how to write a letter of recommendation, when to say yes to commitments versus no, something that we all struggle with, I'm sure. And I think it's a lot of useful information that, and a nice read if you're interested. And that's at her lab webpage. And so with that, I'm going to introduce her seminar. She'll be talking, the title of her seminar will be, "What does cooperation and conflict in microbes have in common with animal social behavior?" And so Joan, if you want to go ahead and take over, and thank you for being here.
Hi, everybody. This is a kind of scary audience. What can I say? All right, let's see if I can figure out how to share screen properly. So, does that work? That's good. Yes?
Yes, I can see it well. Thank you.
Okay, great. Yeah, so I work on microbes, and I think it's kind of tough to make them as exciting as organisms you can see. But there's things you can do with microbes that you just can't do with other organisms. And so we've really had a lot of fun with them for the past few decades. I will say that one of the things that makes working with microbes from an animal behavior perspective is, as far as I can tell, all of the theory that has motivated my deeper understanding of microbes has come from people who work on animals that you can...
Sorry, Joan.
Yeah.
I'm sorry for interrupting, but we see the presenter's view of your presentation and not like the whole slide.
Oh, thank you. All right. Let's see, I'll do a stop share and then share screen, and it asks me which desktop. Is that better?
Yes, that's it. Thank you very much.
Okay, great. Okay. So yeah, the theory comes from people watching animals, and a lot of the microbiologists that didn't have the wisdom to study animals they could see first often made honest mistakes as to what was cooperation and what was conflict. Whereas my first view was, honestly, I have no idea what's cooperation and what's conflict because these things are too little. Let's see if I can advance this. So I'll sort of start from the basics. That group living, most simply viewed, takes two forms. This is just the very most basic. Leave out sex, leave out all of that. You can have aggregations made up of unrelated individuals. Everyone attempts to reproduce. Benefits accrue to all, but unevenly. They will fight over them. And here you have things like geometry of the selfish herd. Benefits can come at different life stages, and there can be very complex interactions. Then the other kind has to do with clones and families, where individuals are related. And this sort of changes the game because it means that you can have altruism where all don't have to reproduce, and the benefits can be cooperatively unequal because of the shared genes. So when you're looking at an organism, I would say one of the first things you have to know is which kind of group are you looking at? What kind of sociality are you looking at? Are you looking at aggregations where everyone is different, or are you looking at clones where you can have cooperation due to shared genes through kin selection? And I'll just let you know how I think about this, which is that nearly all forms of altruism are really from clones or families and not from aggregations. And aggregations don't even actually lead to the other, as one can see with the colonial nesting wasps that haven't resulted in the social wasps. So one thing about this kind of multicellularity, if you think of it that way rather than as a larger organism, is that it almost always goes through a single cell bottleneck. So for animals, it would be your body has, we were all, hard as it is to believe, once a single cell, and the same is true with other things. So if altruism is easy to obtain in clonal organisms, that means that really, if you can find it in non-clonal organisms that originate from aggregation, you might learn something that you didn't expect. You'd expect more conflict, less altruism. Perhaps if we're looking at the origins of multicellularity, there would be less elaborate forms because of that conflict, and increased relevance of the theories having to do with aggregations, perhaps less relevance of things like kin selection. So I'll tell you a little bit about this microbe that I work on. It's an amoeba, it's a protist, it's a eukaryote, not a bacterium, so it's got normal sex. Actually, it has really kinky sex, but it's not really part of the story. You'll have to ask me. So what happens is that tens of thousands of cells aggregate into a single body, and that's sort of their main social stage. They're in the amoebozoa, which is sister to animals plus fungi, diverged about 500 million years ago. And they're common in the soil. Dictyostelium discoideum tends to be in forest soils in the eastern US and eastern Asia. There's about 200 species. Probably there's an order of magnitude more if they get looked at properly. They're haploid through their amoeba stage, which makes genetics easier. They're predators. So most of the time they divide by binary fission, just like any good amoeba should. But then there's the social stage, which is different and which brings us to some of the interesting altruism questions. So here they are munching away in the forest on the bacteria. And so this is its main interaction with bacteria, which is predation. So for those of you who work on real animals, just imagine that this is a cheetah and this is a gazelle, okay? Because it's really exactly the same. So munches it down, typical predation. But then when they run out of food, they aggregate, go towards cyclic AMP. This pipetman tip is releasing cyclic AMP. And so they're moving to it, not only just moving, but also chaining along. And then they form a slug that's actually visible, and that moves towards heat and light, like this. So I was really honored that the Animal Behavior Society elected me as their president at a time when I was working on this organism, and this was the sole behavior that my organism showed that could really be viewed as a behavior. And we've done just about as much with this single behavior as one can. Anyway. So then once this slug has moved to where it wants to be, if a slug with no brain could be said to want anything, and then it forms a fruiting body in which about 20% of the cells die to lift the others up. So you could view the stalk as the soma and the spores as the reproductive tissues. So here's a little summary, a cartoon of this interaction. So they spend most of their time in this stage in the top right, eating and dividing. Here I've got two different clones colored yellow and blue. They aggregate, form a slug that ultimately forms a fruiting body. And this one shows essentially the yellow losing to the blue and making the altruistic stalk. So the big question we had to start with is this what happens? Is social behavior in Dictyostelium more like these aggregations or like these clones? Do they have a way of becoming clonal even though they don't have a single cell bottleneck? So one of the first things we did was to get a collection of wild clones and label some of them and not the others and mix them together and plate them out. And here you can see the fluorescent ones and the non-fluorescent ones. These would be the spores, not the sterile fruiting bodies. So the question is simply, if you mix them equally, does one win over the other? And it's a bit of a complicated figure, not worth explaining, but the answer is that in the handful of wild clones that we started with, we found that in half of the 2-clone chimeras, one clone contributed more to spore than it did to stalk. And so it was essentially cheating the other clone. So we thought, okay, we're in the game. If this didn't happen, maybe I'd still be working on wasps, but it did. So we then asked the basic question of what determines who wins and who loses. And so for this, we then again turned to our knowledge from animal behavior, that if you look at a competition, you could have strength, weapons, as we heard about last week, barriers, could be timing, first strike, last access, information, or numerical advantage. And some of these we were perfectly capable of testing in Dictyostelium discoideum. So we first looked at sickness. So here we're actually taking a genetically uniform background. This is a clone that's commonly used by the cell biologists. And we simply mixed it with labeled clone mates that either were grown up with little sugar or in a pH that was more acid than they really liked. And we found that these stressed out cells, starved or stressed out cells, did contribute fewer spores in chimera than did the wild type. So strength and condition do matter. We then wondered if the first to starve, the ones that first set out those cyclic AMP pulses, I totally thought they'd be altruists. It's kind of like a game that someone's trying to get you to play. Why would you even play it if they were going to then take advantage of you? So we asked that question, and we actually found that the first to starve were more likely to become spore than stalk. And then we realized this had been shown before by a molecular biologist who used a gene knockout that inhibited aggregation, put one aggregation initiator, which then was the first signaler, and showed that it became spore. So this is a nice, really solid support of what we found. And it also points out one of the joys of working with Dictyostelium discoideum, which is that there's a very large, very friendly, wonderful community out there of cell biologists, developmental biologists, biochemists that work on this organism, and they have annual meetings in most times. And they're just really nice and helpful. And I just can't tell you how wonderful that's been. All right, so far, I always have a short attention span, so I like to give lots of summaries. So for those of you who are checking your email, you're not behind. So we've shown that a soma can develop even with aggregative multicellularity. In this case, it's the stalk, only 20% of the cells. We've shown that some wild clones cheat others, contributing less to the stalk. We've shown that winners are stronger, raised on more glucose, have less harsh conditions, and that they're the first to starve, which you might say they have earlier information and so prepare for competition. So I'd say so far a behavioral ecology approach to this system has been good. But I haven't really addressed this key question of, is Dicty more like an unrelated aggregation or a clone? I mean, they do aggregate, but are they aggregating with non-relatives? So to figure this out, we actually had to go to the field and find wild fruiting bodies. And this was our first attempt in the early 2000s. This is Rich Kesson. He was the Associate Dean of Columbia's Med School. He's a hardcore biochemist, could not believe we were going to do field work on Dicty or that we would find these things, and invite him himself on the trip. And he's been a wonderful mentor to us ever since. And indeed, we did find wild fruiting bodies. Though we were told it would be impossible, we knew where to look. And that is where there might have been tons of bacteria, but were no more, which was deer turds of a certain age. So we found lots of these, and grad student Owen Gilbert picked up 88 fruiting bodies from 25 dung piles and found that overall they were clonal. There were some chimeras, but overall they were clonal, and relatedness could be estimated as about 0.86 or very high. So they're mostly, but not entirely, clonal. So that answers that question. But one of the joys of working on microbes is that we can then take this another step and say, well, what if relatedness weren't so high? Can we just knock down relatedness in experimental evolution and see if we can see what happens? And so Jenny Kudzel-Fick did this. And the way we did it is a little counterintuitive, and that is that we just started with a single clone, all genetically identical, and then we picked up 1 million spores and plated them on the next plate, and kept doing that for 30 rounds. So that they started out clonal, they'll stay clonal. But in fact, they won't, because we're doing 1 million cells each time. So any mutant that arises will not be with others like itself. So if it is a cheater that's good at cheating but bad at something else, it could still proliferate because it's got all these other guys around it. So we were just relying on random mutations and the fact that we were moving 1 million cells each round. And Jenny did this with 24 lines. And so what she found, this is a little challenging to look at, but what she really found is that 18 of these 24 lines showed significant evidence of cheating the ancestor. That's the ones that are down here where it's percent change of the labeled ancestor. I probably should have switched that around. And then if you look at those lines, you can see that obligate non-fruiters who totally lost the ability to form fruiting bodies also occurred in most of the lines. And in some of them were even so common that they were close to crashing the population. So it turns out this was easily evolved when they're surrounded by wild type fruiters. So one thing about our work with microbes is we've tried whenever possible to truth experiments with what goes on in the wild. And so in this case, we wondered, okay, if there's low relatedness in fruiting bodies, we should be able to find these non-fruiters in the wild. And so we looked and cultured from clonal isolates. This is also Owen Gilbert's work, 3,316 isolates from 95 wild fruiting bodies. And every single one plated out clonally was perfectly able to form a good fruiting body. So what this means is that it's actually really important in nature to form fruiting bodies on your own. So those low relatedness conditions are unlikely to be common in nature. Okay, so if they have such high relatedness, we can then ask, how is this maintained? Well, it could be from the structure of the environment, or it could be because of active sorting mechanisms based on kin recognition and discrimination. And again, these are ideas straight from animal behavior. So we went to the wild. We used straws to sample the soil. I suppose this is as good a time as any to, that's my husband Dave Queller's hand, and we do all this work together. It makes it so much easier and more fun. And he's really quite a brilliant theoretician, which also really helps. So anyway, so we collected what we thought might be on the scale of amoebas, a 25-millimeter transect at Mountain Lake Biological Station, taking pairs of samples. And then we cultured them and isolated fruiting bodies from these soil samples. And so here the red is the number of isolates and the blue is the number of genetically distinct clones. So you can see that we got lots of isolates. We got genetically distinct clones in these tiny 1/5 of a gram samples. But so, you know, this was a meter-long transect, and then within the little samples, we grew them up. We didn't have a good idea of the fine structure. So a former postdoc in our group decided to really dig into that structure. And so we started by simply plating out 2 clones 5 centimeters apart, and then watching as they grew together and overlapped what the zone of mixture would look like. And so what he found, these are 2 different mixes. So these are, again, the same clone, but one's labeled, that there was a very narrow region where there were any mixed clones at all, that it really tended to be one or the other. And so on the plate, this is what this looks like. So these are all red, these are all unlabeled, and there's just a few mixed along the margins. So growing out from a center, you're going to have narrow boundaries. But then Jeff wondered, well, what are the distances between clones likely to be in nature? So he went back and looked at our samples and at other people's and found that on average, the distance between soil samples was quite narrow, averaging 4.5 millimeters. So this is evidence that structure in the soil alone can result in high relatedness. Oh, and Gilbert, then, you know, you can tell we're really kind of worrying this to death, right? We really want to understand the structure. So Owen then decided, well, he would do it in the lab. He would put agar on the bottom of these little beakers, soil on top, and then mix them, clones densely together, and then genotype them, lots of them. And so when he did this, he found that there was indeed some kind of so we're assuming here that there was no structure because he started with so many. And so he found that there was indeed a little bit of increase in relatedness within a fruiting body. But here it's about 0.05 R, 0.049. So it's a really fairly trivial level, but it's real. So then we thought, well, what might this be based on? And so we turned to two of our then colleagues at Baylor College of Medicine, Gady Shulsky and Adam Kuspa. They said, oh, it must be a cell adhesion gene. And then they even had a guess as to which gene it was, a gene they had called lag genes, but when they realized how cool they were, they just switched their names to tiger genes, something that with animal and microbial species nomenclature would be impossible, but they just did that. So they said these tiger B1 and C1 could be involved in recognition. They're extracellular, one binds to the other, so they'd be exactly the kinds of things that could do that. So again, truthing this with collections from the field, we sequenced about 20 strains from different places, and then we looked at these genes and their variation. And to our surprise, if you look overall at number of genes, we have the whole genome, and nucleotide diversity, these are tiger C1 and tiger B1 were two of the most variable genes in the entire genome. And the other most variable were others in the same family. And so I won't go into all the details of showing how it seems like these are switching in and out and maintaining high recognition. So we then thought, okay, well, it's those genes, let's prove it. So that would take finding a uniform background and then knocking out the tiger gene and putting in another tiger gene like this. So here we have C1 and B1 in this clone and C1 and B1 in that clone. So they should be the same. And then here we have a different gene. So these should be different on this side. You know, we're field biologists, and Gady said he could do it. And, you know, we had a big grant together, and the years went by and they went by and they went by. And we'd always ask Gady, well, how's it going? We really want to know the results of this experiment. He'd always say, Joan, it's going just fine, but it takes time. And it actually took the grad student that did this about 3 years to do this simple replacement. I think now with CRISPR-Cas it'd be a lot faster. So what I'm going to show you here is we've labeled, or they've labeled, only about 22% of the gene of the clones so that we can see them. 2% of these red and 2% of these green, and likewise here. So this is the control side where they're the same. And then this is the experimental side where the reds are actually different from the greens. And I'm going to cry if this doesn't play. There it played. So, what more proof do you need than that to say the tiger genes are causing them to sort and to separate? I mean, we have the control, we have the experimental, all the rest of the background is the same. I said to Gaddy, yeah, that looks great, but that's early in the social stage. What do the fruiting bodies look like? And harder to get a clear answer out of him on that, but of course we ultimately did. And the other reason we were a little quizzical about this was that we had Owen Gilbert's result of showing that the sorting only came to a 0.05 increase in relatedness. So Owen then showed us this video, and this is at a later stage from that earlier one. And this is 2 genetically different clones, none of that fancy gene replacement. It's just AX4 labeled with GFP and a NC2075 wild clone. So one clone is this dark and the other is the green. So just take a look at this one. So here what you're seeing is some are dark, some are mixed, some have some level of sorting. If they crawl over each other, they sometimes stick on. It's kind of like if you went up and shook hands with your friend and then your hand attached and your arm came off at the shoulder, that sort of thing. But so while there is some sorting, as you can see, there's also clear mixing. So this complicates the picture and really makes us ask, well, why would you even bother to have genes for kin recognition, important genes that are the most variable, therefore highly selected in the entire population, if they don't even bother to use them for sorting? This just seemed ridiculous to us. So then we thought, well, maybe they're used to fight other clones when they're together. So maybe they're not using them for sorting at all, but they're using them in that battle to not be stock. This is an evolutionary question, will not have escaped your attention. And so for this, we really need different techniques from the ones we've used so far. We can't simply label clones and see what happens because we want to know what evolved. And so we took a genomic approach and looked at the genes that have evolved to be active when different clones are together in chimeras. So this is Sujin No's work. She's currently on the faculty at Colby College in Maine. So we took 2 clones, 1 and 2. We had 4 different pairs, and then the chimera. And so this is that early pebble stage, which is right when Gaddy's video showed the maximal sorting. And then we simply asked the question, maybe genes are changing expression in chimeras. So we thought about it kind of like if you see an army in peacetime and the men are marching along and they're forming grids, and then they turn and maybe someone's blowing a bugle and they're holding their guns at their sides, maybe across their chest. You would have no idea what's going on compared to in a battle where they're doing completely different things. Yeah, if wars are ever fought like that. But anyway, so we wanted to know about the genes. So we identified genes that were over or underexpressed in chimeras relative to single clone, to pure clones. And to look at this, we used a measure called alpha, which we actually think is very underused in molecular evolution studies. You should all be using alpha for looking at this sort of thing. And it's the proportion of non-synonymous substitutions that are adaptive. So it's simply 1 minus the proportion of polymorphic non-synonymous over synonymous genes over the substitution non-synonymous over synonymous. So under neutrality, alpha is 0. And then with adaptive evolution, only d sub n increases. So alpha will increase. And so here, plotting alpha, this is the genomic background of all the genes that we looked at. And this is just the plot of the alphas of all the genes. This here is all the genes that were turned on, were expressed differently in chimera. And you can see that the alpha is higher. This is the genes that are upregulated in chimera, which is kind of what we were looking for, because we expected things to be turned on, but they could also be turned off, and the downregulated were also more highly expressed. So we then thought, okay, it seems that what's going on here is that maybe they're using these tiger genes to tell them whether or not they should be marching on the field and blowing their trumpets or getting down on the ground and fighting because there's actually an enemy present. So we're kind of really proud of this result. It was not easy to come by, and it answered a pretty important question to us. So just in wrapping up, I'd like to point out, in working with microbes, what a broad variety of tools and techniques you can bring to the questions. We've collected clones from the field all across America. We've done lab experiments where we've mixed 2 clones together and seen what happens. We've done experimental evolution where we've actually seen if an evolutionary condition, in our case low relatedness, resulted in the kind of change that we expected, and found that it did. We've, with collaborators, knocked out genes and replaced them to see if they're causal in what we're looking at. We've used genome sequencing to identify polymorphism in tiger genes, and then looked at expression in chimeras. And above all, what we've done all of this work in collaborations, either with people in our lab or with other labs. We've learned a lot from the community. We've been able to do things that are really hard to do in animals. And I think that mostly we've tested theory from animals, and it's really been a great amount of fun to do this work. So what did we figure out? Again, just the quick wrap up. We've shown that a soma can develop even with aggregative multicellularity. In our case, it was the stalk, which is only 20% of the cells. We've shown that some wild clones cheat others, contributing less to the stalk. We've shown that winners are stronger. They were raised with more glucose and less harsh conditions, in this case, low pH. We've also shown that winners were the first to starve. They had earlier information that the aggregative stage was coming and battles might be necessary. We've also shown that relatedness in wild fruiting bodies is high, and this was from collecting wild ones, something we were told could not be done. And we've shown that if relatedness is experimentally lowered for new mutations, the ability to form fruiting bodies, stalks, and spores, the whole business is lost. We've shown that high relatedness is mostly achieved because clone mates are what is nearby. We've shown that the tiger genes have can have a slight effect on relatedness, but that they're not the main thing for generating high relatedness in fruiting bodies. And we've shown that genes expressed in chimeras show rapid evolution, which is a signature of conflict. So I hope you've enjoyed this dip into the microbial world. I feel like we've worked on these guys for 20 years, but we're just barely scratching the surface. We do a little bit feel that we've certainly collected the low-hanging fruit for social evolution and have moved on into looking at mutualism with bacteria that Dictyostelium discoideum has, and also at predator-prey interaction. So we've diverged from this. I'd be curious if you guys see cool social evolution things we still could do. I'm sure there's plenty. But with that, I'd like to take any questions. Thank you.
Great, thank you, Joan. Thank you for a wonderful talk. Just give me a second here, just to remind people, excuse me, in the question and answer, please put your, if you have a question, please throw a question mark into the chat. And Joan, could you stop sharing your screen just for, thank you. And when you have a question, please, we're trying to build a community, so we ask that you introduce yourself. You know, tell us your name, a little bit about what you're doing or where you're from, and hopefully we can have a good discussion. So any questions for Joan? Okay, David Fisher, please go ahead.
Oh, hi. Yeah, that was fascinating. So my name is David Fisher. I'm a research fellow at the University of Aberdeen in Scotland. So I think I read this in one of the papers you recommended, but is there selection on some kind of the fruiting body traits, like the height of the stalk or something like that? And is there also selection on any traits of the slugs? So is there selection on the multicellular aggregations they form?
Well, I'd say there must be, right? People have looked at selection for higher stocks, and we've looked at selection. We've also seen that conflict will between clones. If you have two different clones mixed together, you will have shorter stalks. So yes. And as far as the slugs go, again, it seems like they move towards heat and light, and it's got to be important to form the fruiting body at the best place. But yeah, those are kind of hard to work on. So we can show the impact of chimeras on them, but hard to work on in the wild.
Okay, thank you.
Okay, our next question comes from Miles.
Hi, I'm Miles Machinsky. I'm a first year's master's student in Lauren Hayes' lab at the University of Tennessee at Chattanooga. My question for you, Joan, first of all, thank you so much for the talk. It's very great. My question for you may not be the sole focal point, but I think it would help me understand. I got thrown off, and also maybe I'm just interested in learning more about the kinky sex of the amoebas, but I was interested when you showed the life cycle in the formation of the chimeric fruiting bodies. I was interested in the ploidy at each stage. So thinking about how these chimeric aggregates can form into one fruiting body, I was kind of interested if between different colonies or different types of amoeba cells in there, are they mating together and creating those haploid spores, or are they diploid spores? I'm trying to relate this to my background in studying fungi, so I don't know.
Okay,
exactly.
So thanks for asking. I love to talk about sex. Anyway, so the entire life cycle was the social life cycle, and those are haploid amoebae. They stay haploid. They don't eat. There's no cell number change, maybe at most one division. So that entire cycle, everything was haploid. Yeah, so one little side on that is if you look at the mitosis cycle, Dicty has a really short G1, and it seems like it's kind of like musical chairs. You don't want to be a tiny cell and lose out if everyone starts starving, because then you'll become stock. So they immediately start synthesizing DNA. So I'll take the moment to talk about sex, if you don't mind. So the sexual cycle is just awesome. It forms, it's also under starving conditions where it's wetter and darker. I am not making this up. And has a different, I can't remember ever if it's more or less calcium in the buffer, but there's a difference. And so what happens is, okay, so we know the genetics of sex. So there's a mating type. There's 3 possible mating types. They can all mate. Any 2 can mate. So wet, dark, starving. They start to aggregate also to cyclic AMP. And then the first 2 that are of different mating types that encounter each other fuse and form a diploid cyst. Then they start letting out more and more cyclic AMP, and more other cells start coming in, and they eat them. So it's like an orgy where everyone's invited. The first 2 have sex and they eat everybody else. They consume them. And, you know, this sounds like, you know, some terrible nightmare thing to have happen. But the ones that are being consumed, as they've aggregated to a big enough number, actually start building a wall around themselves. So they're like in a seraglio. So they're agreeing to be eaten. And so then this one diploid thing that's had sex eats all of these other cells, thousands of them, becomes a giant cell, which then recombines, and then it's called a macrocyst, and then ultimately it hatches and puts out all these little haploid babies. Now, who would not want to work on this? Yeah, right? So we've had one grad student work on this, but the problem is that you can't get those macrocysts to hatch in the lab. And we've tried lots of different things. Other people have tried things. I gave a talk. I don't talk about the sex that often, but I gave a talk where I did talk about the sex, you know, what we'd figured out about sex in, I think it was in Nebraska. And a woman from the audience actually came forward and said she was one of the grad students with Kenneth Raper who first worked out some of this stuff, and that sex was so impossible that she actually got out of science. Not really, she started teaching high school, but anyway, so that's the tragedy of Dicty sex, the glory and the tragedy, more than you wanted to know.
No, awesome. That's cool. Thank you. That helped a lot. And the macrocysts, just to add on really quickly, I'm so sorry. Are they dormant, or are they still kind of, I guess, active, but they just aren't hatching and releasing those spores?
They're dormant.
Okay.
They're
Cool.
very dormant. Yeah.
Thank you.
Sure.
Okay, our next question comes from Tim Caro.
Hi, my name's Tim Caro. I'm at the University of Bristol. And I just wanted to ask you, Joan, what's the advantage of the height of those stalks? Presumably, if you're in a fruiting body, you want to be as tall as possible for reasons I don't understand. And then that's going to create additional conflict between the guys who are in the stalk and the guys who are in the fruiting body.
So we've done a little work on this. The taller the stalk, the more likely you are to be picked up by a passing insect or something. So these spores are actually, they don't blow, they're sticky. And so we've done experiments without stalks and with stalks, and the Dicty gets transported. We haven't done it with high and low stalks, but the idea is that they simply are more likely to be transported to a more favorable place to live, is our thought about it.
Okay, thanks.
Okay, our next question is Karsten Schraden.
Yes, hello. Thanks for a great talk. Fascinating. I'm Karsten Schradin from the EPHC, CNRS in Strasbourg, France, studying striped mice in South Africa. And I have to apologize for my ignorance. I mean, it's so fascinating, but I didn't understand one of the most basic things, and you might have explained it more than once, maybe even to Miles' question, while I still was trying to understand it. Now, you have these single haploid cells, and then they're starving, and then they form these fruiting bodies to produce spores, which probably are more resilient to the environment. So why do they need to build these fruiting bodies? Why can they not just say, okay, I'm starving, I build a spore to survive? So why is this phase necessary, and what do they get out of it, and how does this work?
So I think that the reason they build the stalk, in which 20% of them, so they become spores, right, or stalk, and we think it's for dispersal. So they build the stalk and then they're dispersed from the top of it more effectively. There's other genera of Dictyostelids that can make what we call microcysts, i.e. they can make a fairly hardy spore without the social process, and that they have either or. And the spores formed by microcysts are not as hardy as the ones formed through the social process. So we think they're probably also getting some extra membrane, something or another, from the social process. But yeah, it's dispersal is what we think.
So it's dispersal, and you think they take up some resources from the cells that do not contribute to becoming spores.
Yeah, but mostly they get the stalk from it.
Okay, thank you.
Next is Michael Taborski.
Yes, John, I would like to understand how the cells, the single amoeba coordinate to form the slug and then also to move in a certain direction. I mean, this is not a trivial task, I think, and I could imagine that there is also some kind of conflict because the different cells have different information from the environment, and perhaps also different statuses, I mean, different states of starvation and things like that. So do you have any idea about how this is coordinated and how it moves in a sort of coherent direction and then also changes to form the fruiting body?
So the way it works is they all aggregate to the cyclic AMP, and then the middle of the aggregation starts to form the front of what becomes the slug. So it kind of goes up and then it collapses. And the front of the slug, the few cells at the front, are where things are organized, and those move. So the front moves and the rest of it just follows along. And I think the cell biologists have figured out some stuff about the actual mechanisms for how that works, but I don't know them.
So the front is defined by, as you said, the middle. So it's the region where most where you have most neighbors around you, or something like that. Is that correct?
Well, in the aggregation, they all come together and then they sort of go up and then flop over. So yeah. Yeah, it's sort of the top of the mountain, then becomes the front. And those are also the cells that form the stalk are in the front. So there's different genes that are expressed in the front, and there's ways to figure that out. But yeah, I don't really know the exact mechanisms for it.
Okay, thank you.
Next question is Baptiste Zagdui.
Hello. Hi, so I'm Baptiste. I'm a PhD student at the University of Göttingen, working on primates aging and with a focus as well on gut microbiome. So thank you very much for the talk. I was especially interested by maybe the last results you presented. So when you have this differential activation of genes in response to the signal captured by those tigers proteins, I was wondering, like, it almost reminded me of an MHC kind of behavior on immune cells, and it triggered me to wonder, do amoeba cells actually have something that would kind of look like an immune system that they use in those conflicts, or is it more a competition that would remind us of predation, like eating each other? And given the colleagues that you work with, have you ever talked about this similarity with those MHC proteins that we see in eukaryotes? Thank you.
So yes, they are like the tiger genes are like MHC genes, and they're highly variable and all of that. We haven't seen any function, any MHC-like function. What we have seen, and we've worked on a little bit, is colleagues again at Baylor, Adam Kuspa and Gady Scholsky, discovered cells in Dicty they called sentinel cells, and they're only in the slug stage, when the slug is actually a little tiny organism that crawls around. These cells have lowered adhesion, so maybe that would be tiger involved. And so as the slug moves, these cells kind of get dragged to the back, and as they move through the slug, they pick up any bacteria, any waste products. So it's kind of like a kidney, a liver, and an innate immune system all wrapped up in one. And so that's sort of the closest I can say that Dicty has to these things.
So if I understood correctly, it's almost like a specialization of certain haploid cells that turn into immune-like.
Yes.
Okay. Well, that's fascinating, almost like insect specialization. Okay, thank you very much.
Yeah, sure.
If you have questions, please put them in the chat. Keep them coming in. Our next question is from Jinju.
Hi, I'm Jinyu from University of Strasbourg in France. I'm a Master 1 student, and I'm currently working on marsupials social organizations in Karsten's lab. And my question is, have you found any differences in the proportion of the stock and the food in different sites of aggregations? Because I remember the picture you show, like if I'm not making any mistake, in the picture you show, the size of the food are quite even, but the height of the stick are not. So does it mean that, like, bigger aggregations with higher stick, they actually have less proportion of individuals that can end up in the fruiting bodies?
So I guess what you're asking is, if you make a fruiting body from 1,000 cells, will it have a different number of cells in the stalk, a different proportion in the stalk, say from 1 million cells? And that has been worked on. It's tough to count the cells in the stalk, and there's people have done it in various ways, but I think there's probably a sweet spot in stalk size, so that if the fruiting bodies are too small, they'll have more cells in stalk than spore, and then they'll get to a point where larger ones have more cells in spore than stalk. So when we say 20%, that's just kind of a rough average. So yes, there is a difference. And so, yeah, you would think that they'd want, be selected to be in a fruiting body that reached the highest height at the minimal cost to making stalk cells. Does that answer your question?
Yeah, so does it mean that if you are in a bigger aggregations, that means you are facing higher, more difficult, higher competitions to reach the fruiting bodies?
Well, so the fruiting body is the stalk and the spores. And so sometimes from a really big aggregation, you can have multiple slugs go out. Yeah, getting at this stuff is tough, and it's hard to say. And we even wonder, in the lab where we put them on a plate full of food, exactly what the relevance of it is in the wild. So that's partly why we haven't pursued it too much. But I think for sure there's a story there. I'm just not sure what the answer is.
Okay, thank you.
Sure.
Okay, our next question comes from David Fisher.
Hi, yeah, me again. So I have a more like a quantitative genetics background, and there we think I think about partitioning the variance in this behavior, say how much they cooperate into this percentage from genes and this percentage from the environment, and you showed that there is variation in cooperation due to certain genes and due to the condition they're in. So can you hazard a guess at what percentage of the variation in behavior is due to these different sources? And is there any left over that is explained by something, some emergent property or something else other than this genetic variant and this environmental factor?
So if you're asking about relatedness, how much is due to structure in the environment?
No, more I guess in a population of these slime molds, what percentage of the variation in their ability to cooperate is due to genes, and or ability to end up in the spore is due to genes, and what percentage is due to the environment? Or can you compare the relative magnitude of those two factors? Like you showed the difference the condition makes in terms of exposure to low glucose or the low pH. And can you compare that magnitude of difference to how much difference certain alleles make?
So when we first started working on them, that was one of the questions that we really wanted to answer. So we thought, for example, that clones that came from a dense population would genetically have better ability to compete than, say, clones that came from a less optimal population where they were less likely to run into non-clone mates. We have shown under fairly solid conditions that there are some clones that consistently beat other clones. So as far as, which is genes and which is environment, they don't run into others. The chimeras is a real thing and the competition is real, but I'm just blabbing because I really have no idea. I mean, we have solid evidence about the environmental impacts. Holding the environment constant, we have solid impact of different, couldn't say which genes, but that some clones consistently beat other clones. How we would put those two together in a way that was meaningful in their natural environment is not something we've really figured out. Whenever I give talks, my main answer is I don't know. So I always try to write down the questions and study them, but I never do.
Next is coming from Miles.
Hi, another repeat. So I guess kind of on that same note, even if you don't have clear methodology planned out or an idea of how to assess how much these interactions or differences between colonies are determined by their genetic and their like DNA versus the environment, even if that's not clear, being able to clear that up and see which is more of the driving factor. Is there an ability to kind of assess their genetic heredity, and so seeing specific genes, if those genes being passed down, I guess because they're colonies, it may make it not so, it may make it more difficult, or it's not applicable. But to look at seeing if like the phenotypes that they're expressing are because of their genetics, and seeing like how much of the actual genes that are getting passed down and the alleles that are getting passed down are prompting those phenotypes to be expressed. I don't know if that makes sense or not.
So I talked about the social cycle mostly, and so that they just divide by mitosis and pass on their exact same genes. So if you do mix two clones, one will prevail over the other, and so then its genes will get passed on. So beyond that, I'm not sure if I understood you right.
Yeah, no, you probably wouldn't be able to like calculate the genetic heredity because it is just the same genetic material being passed down. It's not like a mix like you would get between like diploids.
Yeah, but we can do experimental evolution and then see what genes have changed and stuff.
Okay, that's interesting. And then kind of a second one just to add on more and confuse myself probably more, and I got caught up on this then. You said that the fruiting bodies don't disperse spores through wind dispersal because they're relatively sticky. Can they disperse through like rain dispersal?
Rain would just wash them into the soil where they are, and sure, they would flow along.
Okay.
But rain would also disperse them if they didn't make fruiting bodies. So we really think it's, insects and things like that. We had a panicky moment where we started worrying about whether the stalks had evolved before insects, and therefore our whole story was moot. But I don't think that's a problem.
Thank you.
Sure.
Keep bringing the questions in. We have another question in the queue. It's Lizan.
Hi. Yeah, thank you for the very interesting talk. My name is Lisanne, and I'm a first year master student in Groningen. And I was just wondering, and I'm sorry if I might have missed it or misunderstood, but in the end, the tiger genes only partly contribute to the sorting and the high relatedness of bodies in the wild, right? And so another reason that you mentioned or contribution was the environmental structure. So clones being more close to each other. But as I understood it, this also only explains it partly. So Neil, I was just wondering whether you found another reason or mechanism that could explain that high relatedness and sorting within the bodies in the wild.
So we think that the structure does explain it. So, if you have a single spore in a spot, they divide about every 4 to 8 hours, depending on the food. So it won't be long that they will have exhausted the tiny spot and made a fruiting body. So our experiments were designed to get to understanding that, but we're pretty sure that that's the explanation.
Okay, thanks. Yeah.
Next, we have questions from YouTube presented by Cheyenne Smith.
Hi, I'm Cheyenne. I'm at the University of Tennessee at Chattanooga, first-year graduate student. I am relaying a question from Amy Post, and they said, I study human functioning through the lens of Bowen theory, not a scientist, all capital, so forgive me for the novice question. It said, did I hear correctly that a tipping point for conflict is the provocation of rapid evolution? Is the null to increase conflict a stability of evolution in relationship to predictable environment? And they also said this is a rich presentation that they look forward to watching several times to really comprehend.
Oh dear, I hope she doesn't watch it several times. But anyway, I think we were using, oh geez, I lost it. I think that she has the cause and effect backwards, that we used rapid evolution as a signature of conflict, not the other way around. Does that make sense?
I think so, but since it's on YouTube, I'll have to wait and see.
Okay.
I can repeat. So did I hear correctly that a tipping point for conflict is the provocation for rapid evolution? Is the null to increase conflict a stability of evolution in relationship to predictable environment?
So that is a tipping point for conflict, what was that?
The provocation of rapid evolution.
Oh, I guess what I would say is no, that rapid evolution is a signature of conflict, because if you're evolving against another active player, you try this and then they learn counter adaptation, so you try something else. So a signature of a gene that's been important in conflict is that it has evolved rapidly, is how I think of it. Maybe that makes sense. She can shoot me an email if she wants.
Okay, thank you.
Sure.
If there are any other questions, please put them in the chat. So Joan, I had a question. I maybe just looking for more of an explanation. And I apologize if you spoke about this. I read about cooperation in foraging. Am I correct that they cooperatively detoxify toxins from E. coli? Can you elaborate on that a little bit? Or did I miss that? Was I totally off on that one?
Do Dicty cooperatively detoxify E. coli?
Yeah, are they, is there any cooperation in the foraging? And I apologize, my brain is melting over here.
Well, so no, that's totally a fine question. So actually, that's a direction that our future work is going to be going in. I'm thinking that what you're referring to is a paper where Suzanne DeSalvo was the first author, and we tried Dictyostelium discoideum on eating a whole bunch of different bacteria. There'd been a previous paper looking at that. And what we found is another great old chestnut from animal behavior, which is allele effects. And so what we basically found was that the relative numbers of bacteria and Dicty can really matter. And so if you start off Dicty on a really dense lawn of bacteria, they may often fail. Whereas if you start Dicty more common, they will do better. So that implies that, yes, they must have some sort of joint ability to, they must have some joint ability to beating the bacteria, but yeah, we don't know exactly what it is.
Thank you.
Sure.
We have a question from Maren.
Oh, thanks for the talk. My name is Maren Hook. I'm from the University of Derby in the UK. I've two questions, probably both short ones. So you had this video where you had the green clone and the black clone, and they fused together. And I don't know whether it was an artifact of the film or whether it was a real pattern. That it seemed to me that in some cases there was one of the clones in the inside and then sort of like this, and then the outside, like a shell, was the other one. Was that just the filming or was that a real effect?
We haven't analyzed those in detail. I think it could be that as one goes over the other, that there's a surface something that keeps them from fusing, so it might envelop the other. I wouldn't say that they stay that way, but yeah, I hadn't noticed that. That's really good that you saw that.
There were some cases where it was like this pattern, and some where it was the green ones at the front and the black ones at the back. So they always seemed to me as if there was a pattern. That's why I was asking. But
yeah, I think you're right that sometimes it was really clear that, you know, one or the other was at the back, but sometimes they would crawl over each other and stay different. Other times they would crawl over one and just absorb it into themselves. Yeah, the main point I had with that is just that even if they sort early on with those tiger genes, later they will mix up. And then, of course, that was on a plate, and we don't know what it would be like on, you know, in soil.
Okay, thank you. And the second question was about the tiger genes. So did you, because you said you didn't find much of an effect of the tiger genes in terms of sorting them properly, so, or, well, to at least to change the genetics in an appreciable way, or the relatedness. Could it be that there is actually another gene that you haven't, because you were successful right at the start with this gene, that actually you missed a more important one?
That's always the case, isn't it? So the tiger gene, so I showed you that one figure showing how variable the tiger genes were. We looked at the other tiger genes, not B1 and C1, and they didn't seem to cause the sorting. I just think that overall, when we mixed two clones, it didn't seem like there was a big genetic component at all in terms of what ended up in the fruiting bodies. So I think we know what all the cell adhesion genes are, and the others tend not to be super variable. So there was one we were pretty interested in. What was it called? I can't remember. But yeah, none of the other genes had the kind of variability that you would need. So no, I don't think there's another gene lurking out there. You know, it's
okay, thank you.
Sure.
We have another question from Jingyu.
Yeah, hi, me again. And so I just came up with another question. Like, have you ever, so have you found, like, do they have other, no, is this fruiting body strategy is it the only strategy they have to deal with harsh environment, or is there that they have other strategy to survive?
Well, there's the sex strategy that I talked about. But yeah, I mean, they form the fruiting body. At the top of it, they have hardy spores that can last for years or even decades. So yeah, so that's their one game, or their two games, or this macrocysts and the spores. Yeah.
So do you know, like, when would they choose to do the fruiting body, and when they will like choose not to aggregate, but to like you said, to choose the sex strategy?
Yeah, so it's well known, and it's wetter, it's dark, and then there's different electrolytes in the buffer, different phosphate levels or something, and it's well known. So people that want to make macrocysts in the lab have no trouble doing it. Also, they have to find the opposite mate, one of the other two mating types. So if they don't find the other two mating types, then they can't have sex. And they do have, so we've also done just genetic screens where we've looked at variability in the populations to see if, you know, they ever have sex, and there's tons of recombination. So they certainly do have sexual recombination fairly frequently.
Okay, yeah, thank you. So I actually was also thinking that, does it, as you said, that fruiting body can last for a very long time. So does it mean like when they sense the environment is getting very harsh, and they would prefer to do the fruiting bodies instead of the sex strategies? Like it's a better, like fruiting body is a better way to deal with harsher environment.
I don't think they can really pick. I think that as the environment gets worse, as they have less food, the other variables will make them go on either one path or the other.
Oh, okay, thank you.
Sure.
Karsten, please.
Yeah, sorry, I want to follow up on Chingyu's question. Can you be sure that they are not capable of building spores without building these fruiting bodies? Is it possible there's an unknown strategy that instead of joining a fruiting body that they don't like because of relatedness, they build a spore by themselves?
I would say that they can't. I think if they could, the cell biologists would have discovered it. It's also, so Dictyostelium is now recently divided into a bunch of different genera, and the whole genus discoideum cannot do that. And there's other genera that can. So you can't ever say, you know, for sure that something doesn't happen, right? But yeah, we've certainly never seen any sign of it, and it's common in other genera. So yeah, and then I think if I were a cell biologist, I would start rattling off pathways and how they were intertwined and why they totally couldn't do it.
Okay. But other genera, they can do it. So they have the choice to make a spore by themselves that might be not very, might have less resources whatsoever, or to build fruiting bodies.
Yes. Now these other genera tend to make really tiny fruiting bodies, much smaller than the ones of the discoideum group. And those spores do not last as long as the spores from the sexual stage.
Okay, thank you.
Sure.
I don't see any new questions in the chat. If you have one, please post it in there, or if you just want to comment on a part of the research.
Well, it's been a long time. I'm sure everyone's pretty tired.
Yeah, it's been a really nice discussion. Oh, Cheyenne has a question.
I'll ask. I don't know. This might be silly, but is the reason that you guys thought you wouldn't find them, or people thought you wouldn't find them in the field, just because of their size, or is there more to it? I don't really know a lot about this. So.
Well, so yeah, they're little, and some of the cell biologists had looked for them, and then there was another famous person who had made a really concerted effort to find them. And so we were just told we wouldn't be able to find them. And, yet the first old deer pile of dung, we found them. Yeah, it was kind of funny.
Okay, interesting. Thanks.
Well, if there are no more questions, I encourage people to just stay around and chat.